We are still in the same concept of homing, recirculation, hoping that the naive lymphocytes meet their antigenic counterparts, so they will here be primed, and then they will change into memory cells. So this slide explains the concept of homing and recirculation as well. So the process of homing and recirculation is an active process where the attachment proteins that are present on the lymphocytes, they have to meet their antigenic counterparts on the endothelial lining, and then they will bind into those actively. So you can see here those attachment proteins that are present on the lymphocytes and their counterparts on the endothelial cells. So they pass through these stages where the rolling will stop, and then they will pass through the endothelial lining through a process we call it diaphydesis, and then through the cell, then through between the cells, they will pass into their allocated area in the lymph node. So this is again explains the molecular aspect of the process of homing and recirculation of the lymphocytes within the lymphoid cell, and it shows here how those cells they will start to develop new antigens on their surface in response to cytokines that are produced, and then they will stop rolling, they will stop or they will hold, and from there they can pass through the endothelial cell lining. And this process of homing or stopping of the lymphocytes to pass actively through the endothelial vessel, we call it diaphydesis. Yeah, this slide shows more clearly the process of transfer of the lymphocytes within the lymph nodes through the process of the diaphydesis. And so here it shows the rolling cells and how they adhere into the endothelial lining, and then how they develop the intracellular adhesion molecules. They stop, and then they will pass through the endothelial lining into the allocated area in the lymph node, and then they will stop after that. They will home, as we say. So this slide shows the homing process and relationship to the different types of cells. For example, the phagocytic cells, macrophages or neutrophils, they will migrate into areas of inflammation, while, for example, the B and T cells, they will migrate into the area where antibodies will develop, or to the areas of the effector functions of the cell-mediated immunity. So in each area, it depends on the type of cell where it's going to migrate. And this migration usually is mediated through the production of substance, we call them cytokines. So under the influence of these cytokines, and they will migrate into their allocated area. Yeah, this slide will shows these attachment molecules on the cells when they home, uh, and their ligands here on the endothelial cells. The first cells we call them selectants, and then after that we have what we call integrins. So selectants and integrins, and those each one of these selectants has a ligand that is going to attach into their allocated area in the lymph node that will make the cell to hold, and then diabetes is what they will pass through the endothelial tissue. So I think you should know the attachment protein and its ligand here that will make the process of homing and recirculation. And this slide shows more details of these attachment proteins and their ligands during the circulation. That shows that it is an active rather than a passive process. So the first step where we have the primary adhesion, and the first step in the primary adhesion to the endothelium, and then after that here we have the cytokines that are produced to make the lymphocytes to be activated, lymphocyte activation to produce the ligands. And then the third step after that, the secondary adhesion, primary and then secondary adhesion, or the arrest. So the cell will stop here from moving, and the last stage here will migration of those cells, or like as if they are attracted through a process we call it chemotaxis. So the diapedesis, the cells will pass through the endothelial cell lining, migrating into the allocated area in the lymph node. So again, this table shows the summary of the homing and re-homing of the lymphocytes into their allocated areas in the lymph nodes. And here we can find all of those figures in the textbook, which is immunology for medical students where this lecture has been taken from.
كيف الصوت جربتوا؟